Okay, so how we use this monoclonal antibody in medical field, generally in research, okay, in diagnosis, as long as the treatment. So in our syllabus, the example use of monoclonal antibody in diagnosis include the urine pregnancy testing to test for the pregnancy, okay, or in nowadays we talk about the COVID. Okay, so how we test out the COVID-19, okay, by using what we call the rapid test kit, RTK, okay, rapid test kit, okay, RTK stands for rapid test kits, okay, so RTK going to target, okay, to check for the viral antigen, okay, so, and also we detect a disease protein, or sometimes we detect the antibody that's specific to the disease. Okay, so we have different way of doing this. So first of all, let us look at this urine pregnancy test. Okay, now this is the overall example. Okay, you can take a photo to record down for this process, but I won't talk about urine pregnancy test now. I'm going to use a COVID RTK test kit so that you guys understand why you have two lines positive, why one line is negative. So this one is very related to the current situations. So I will go into the detail to tell you guys about this COVID-19 RTK test kit, how the RTK, the rapid test kit, okay, how it functions, okay. Okay, so let us look at this COVID-19, okay, COVID-19 RTK, rapid test kit. Okay, so you put this test kit, okay, you can see that this test kit, put it horizontally, you can see that you have these regions called the sample loading region, where you load the sample. Okay, I will call it sample loading route. So where you're going to load your sample, your saliva sample, and you mix everything together. Are you clear? When you mix everything together. Okay. Now, then you have another windows here. And at the windows here, you can see that they write a T and a C. So T stands for test. Okay, test, whether tested positive or tested negative. C stands for control. Okay, C stands for control. Okay, so either in the result, okay, after we try out, so if you can see that the T and the C here, if T appear one line, C appear another line, so COVID positive. Okay, if you have this T, C, one line at C, like this, then COVID negative. Okay, so hopefully after today you can actually explain to your parents why. Okay, if they ask, if you see no line, or okay, you see one line as T, so it means that this kit actually invalid. So why invalid? Later, when I tell you guys the principle, you will be able to know. Okay? Okay. So now we look at the principle. What is the principle? Now look at this. So if you have, I mean, use this test kit. If you had use this test kit, so first process in the tube. So the tube actually contain, okay, it depends on model lah. Okay? It depends what company. Sometimes they may have the tube, I mean, the reagent tube outside. So this tube contains first, okay, contains the monoclonal antibody. So this monoclonal antibody, okay, this monoclonal antibody contains the targets the COVID antigen. Okay, so let's say we inject this COVID antigen, okay, onto these the mice. Okay, so again the rat. Okay, for example, so we say that this is a rat anti-COVID. Okay, I use this word anti-COVID, right? Anti the COVID protein, basically COVID antigen, but I use the word COVID. Okay, rat anti-COVID antibody. Okay, rat anti-COVID antibody. Are you clear? So means that, but this antibody is special. This antibody, if I draw it out, this antibody will bind to the COVID antigen. So this one will bind to the COVID antigen. Okay, but at the FC region here, this antibody actually conjugated, we say that. Okay, conjugated with the dye. Okay, for example, red dye, it give red color. If the company using the blue dye, it will give a blue color later. Okay, so red anti-COVID antibody. Okay, look at this. Now, what will happen now if you add in your saliva? Okay, saliva sample. Okay, add in saliva. So imagine this person COVID positive. So COVID positive, then definitely they will have the COVID. Okay, green color is a COVID antigen. Okay, so this saliva actually contains COVID antigen. Are you clear? So we mix. So once you mix it, what will happen? So when you mix it together, then you can see that this okay, let me duplicate it. So this monoclonal antibody, or we call rat anti-COVID antibody, now bind with the COVID antigen. Bind with the COVID antigen now. Okay. So what you do? This sample, okay, the mixture now, you load it into this COVID test kit. Right, after you mix it 10 times, okay, you squeeze the boy, then you load your sample. So because this person COVID positive, so the sample actually will form this, okay? Can you see that? Now, capillary reaction take place. So therefore, you put a sample, so sample will, because capillary reactions continue to move. And this side here, at the T and the C here, we don't see anything. Actually, they do have something here. So what actually happened in T and C? So at T, okay, at T regions there. So at T regions, actually, okay, I will enlarge it. At T region and C region. So what actually happened at the T region? At T regions, you have the same antibody, but they are immobilized. Okay, they are the same antibody. Okay, I draw it a simple way. Okay, so we have what? At T region, we have the immobilized, means that we purposely let it stay on the kit there. So we call immobilized rat anti-COVID, okay, antibody. Are you clear? So means that we produce this monoclonal antibody by using the rat. So this antibody actually anti-COVID antigen. Are you clear? How about C? C, then we also have immobilized, okay? But C, we immobilize with different thing. We immobilize. If the first one we use rat, this one I might want to use a rabbit. Immobilize rabbit anti, okay, rat antibody, antibodies. What is this mean? So means that, at control regions here, you will have this. We inject the rat antibody into the rabbit, so that rabbit, because rabbit, this rat antibody, when inside the rabbit will see that it's a non-self antigen. Rabbit will produce the anti-rat antibody. So where are here? Where are them? They are here. So later you see why control always will have the line one. Okay. So what actually happened now? Just now, this sample continued to move. Okay. So continue to move. When it reached the T region, what will happen? Because it's positive, then it forms sandwich. Now, what means form sandwich? Because this is anti-COVID kind of. So means that you will see something like this, guys. The red dye. Yeah, just now this one. You see that? This one move because this is a COVID antigen, right? This is COVID antigen. So means that this one also can bind. The COVID antigen also can bind to the immobilized antibody. And this, the mobile one, carry the red dye. So that's why first line appear now. Can you see that? You can see that first line appear. So when the first line appear, then you should be worried. This time you should start worry already. You can start worry because first line appear. Why first line appear? So sometimes you do not need to wait until 15 minutes. When you can't see a first line, okay, then you have to pray. This kit may be invalid. First line at T, don't have the C. It cannot be if the kit, work well. Because if the kit work well, this antibody, because not all will bind, some of them, there no chance to bind, will continue to migrate to the C region. C region is where these rabbits start to target the rat antibody. So don't care they have the antigens or don't have the antigen. So maybe some of these they don't have, but all of them carry the red dye. So that's why at C, okay, will give us the second red line. Can you see that? This is positive. So two lines give us the positive. So in this case, because of the presence of the COVID, okay, positive, COVID positive antigen, COVID. Now, how about negative? What will happen in negative? Now, is if negative. So now you'll be able to explain to your parents now. Okay, so T and C. So T, you have the immobilized antibody, monoclonal antibody. Immobilized monoclonal antibody. Immobilized monoclonal antibody. Now, because if there is no COVID, okay, so this sample that contains the dye, okay, so without COVID, so means that they're empty. So when they pass through the first line at T, they do nothing because nothing to greet. Can you see that? They are the same, so they won't target each other. So they continue to move. But when they move to the C regions here, then yes, because it's a rabbit anti-rat, as long as rat antibody pass through, they will greet. And this antibody contains the red dye. So when it contains a red dye, so now only one line appear at the C. So it means that the test indicates that negative and it's valid. Because if without the C, we do not know whether maybe your sample move or your sample just stay in the well, didn't move. Right? After 15 minutes, oh, no line, you consider negative. No, it must have one line at C, no matter positive or negative. So this is how COVID test kit actually functions. Okay. Okay, so for urine pregnancy test, you just need to have the clinical sample with analytes. In this case, the clinical sample will be the urine. You straight away, you don't need to mix the urine with the antibody because the antibody already here, the mobile one, right? The mobile antibody is here. So what we test for the urine pregnancy test kit, we test for HCG. Okay, we test for this humans chronotrophins. Humans chronic gonadotrophin hormones. So these hormones only special, okay, only release when girl pregnant. Okay, when female pregnant. Okay, so how long, how much, I mean, the concentration, normally when girl, you have, sexual activities, right, unprotected sex, then this pregnancy take place during, fertile periods, so fertilization take place. So when you miss the subsequent, the next period, and then you realize that you have the unprotected sex, sexual activity, okay, what you need to do, you cannot immediately test it because the concentration of HCG is not strong enough to appear in the urine. But if you eagerly want to know, you can go for blood checking, not this one. Okay, if you want to wait until the urine, this HCG concentration is high enough, you have to wait another 2 weeks after you miss. By right, if you have a very regular period, you miss the periods on this week, okay, by right, you should have the period, then you wait another 1 to 2 weeks, then you test. So these 2 weeks, okay, it depends on whether you want to have baby or not. Okay, if let's say you want to have baby, yeah, you also very, very excited and also worry, okay, but if you don't want to have the baby, this 2 weeks is a hell week for you, because unless you go for the medical checkups, okay, for the blood checkup, okay. So now you can see that this urine, if the urine contains the HCG, the starch HCG, can you see that this antibody bind to the HCG, right? Antibody bind to HCG. This antibody is already inside the kit, not like COVID, you need to mix it. Are you clear? Not like you need to mix it. So you continue capillary flow until the first line. The first line, okay, first lines, they are the monoclonal antibody, they bind to the HCG. So therefore, when HCG bind, it forms sandwich. Can you say HCG actually sandwich between two antibody, and the antibody contains the dye. So give us the first line. But some of these antibody have no chance to bind. They will continue to move. When they move to the second lines here, then you can see that the second antibody, they are the one that target the first antibody. So therefore, you can see that they bind, give us second line. So control line must be there to give us the valid, okay, interpretations of the result. Okay, so now we have the false positive and false negative results. Okay, so if guys, you use this urine pregnancy test kit, you pee on these sample regions here, and you have two lines. Don't tell people that you are pregnant. Say, guys, you can't pregnant. And under healthy conditions, male won't produce HCG. Okay, male will not produce HCG. So you are, in this case, you are false positive for pregnancy. Male, right? For male, so you will be have false positive for pregnancy. But I advise you, or you have to go and see doctor and tell doctor, because testicular cancer can produce HCG. Are you clear? So you get two lines. Hey, I got the HCG. Why? Go and see doctor for male, because testicular cancer actually can result in the productions of HCG. Clear so far, guys? Okay, now, ELISA, you can read about this, ELISA. Okay, so ELISA actually is used just now, as I say that, I won't go into the detail. This is called sandwich ELISA. So we have the capture antibody. So we let the antigens there. So we actually use the biotin label detector antibody. Then we have the enzyme, we give a color. So once we have the color, then you will be able to know whether what we call these reactions take place or not. Okay, so I won't go into detail. You can capture, you can read about it. Says, did you know 12.4? Because for today, I don't want to go into detail about this. Okay? In A2, we're going to learn about this ELISA. Okay, same goes to the Western blot as well. Okay, I don't go into this too in detail until we reach the part. Just to let you guys know, because we talk about ELISA. If you're interested to know more, please go and have a look. ELISA as well as Western blotting. Okay, so both of them, we detect what? We detect specific proteins. Okay, we detect a specific protein. Okay, so you can take the photo of this. And also this one. Okay, this is totally not in our syllabus for AS, but in A2, yes. Okay. Now, we come back here. Deep vein thrombosis. Okay, deep vein thrombosis. Deep vein thrombosis is the formation of blood clots in the deep vein, most commonly in the leg or pelvis. So the mechanism of clots formation typically involves some combination of decrease the blood flow rate and increased tendency to clot. So it means that when you sit still for, you do nothing, okay, you do nothing, and then for a long period of time, then the blood clot will start to take place. Risk factor include when you have recent surgery, older age, active cancer, obesity, personal history and family history, trauma, injury. So you can cause deep vein thrombosis. So deep vein thrombosis can be very severe or serious because the blood clot in the vein can break loose. Can you see that? The blood clots here break loose, travel through your bloodstream and lock in your lung, blocking the blood flow. It will cause a pulmonary embolism. But if this blocked, okay, this embolus detach and then go to your coronary artery or the tiny artery or even the capillary at the heart, it can cause the cardiac arrest. Are you clear? So we need to know where they are. Okay, so we can use the monoclonal antibody, can be used to locate the position of the blood clots in the body of a person through thought to have the deep vein thrombosis. So what we're going to do, very simple. We have the monoclonal antibody. Oh, sorry, wrong. This monoclonal antibody, okay, carry, okay, now in this case, they don't carry dye, they carry the radioactive substances. Are you clear? Radioactive substances. Because no point you give color, deep vein thrombosis. So means that if we detect, we also cannot detect because color won't be shown. Color, the color won't penetrate through your skin. Are you clear? Okay, so we use radioactive. So now this antigen binding site, they bind to fibrin because blood clots have fibrin. So fibrin will bind. So we inject this monoclonal antibody into the vein. So let this monoclonal antibody flow. Because these blood clots, okay, blood clots, we want to find where are them. So this radioactively labeled monoclonal antibody will come and bind. So majority of them will come to this region and bind. So if we use this gamma ray detector, we will be able to pinpoint out where are the clots. Okay, so we can try this, arrange the following events in the correct order. I will disclose the answer later. So first, what we're going to do here is, so a radioactive chemical that produces gamma radiation attached to the monoclonal antibody molecule to make radioactively labeled antibody. So this labeled antibody now against human's fibrin, the main protein found in the blood clot. Okay. So next, so this labeled antibody now introduced into the patient blood. That means the blood vessel, okay, not blood. Okay. Then what we do, as the monoclonal antibody are carried around the body in the bloodstream, so they bind to the fibrin highlights. You need to know fibrin's molecule, means because the fibrin molecule is the blood clots, right? Fibrinogen change to fibrin when they come into the contact. Okay, so the radioactive emitted by this labeled antibody is used to detect where they are in the body. Okay, so how we detect? So a gamma ray camera is used to detect the exact position of antibody in the person body, and then the position of labeled monoclonal indicates the position of any blood clot. Okay, so QSRUPT, very, very simple. We also can use, okay, we can also use this monoclonal antibody for blood group typing. Okay, for example, we have type A serum, okay, so type B serum. So type A serum will have anti-B, type A will have anti-A. So in this case, you can see that type A, because type A only anti-B, so it won't anti-A, so you can see a no agglutination. But if you have anti-A for the type A, can you see that? The type B serums, okay, so type A bloods, you can see that because they have anti-A, so therefore agglutinations take place. So in this case, we know that, oh, this is blood group A. Blood group B, can you see that? AB, then we have both of them agglutinate. So basically, any agglutination indicates that particular blood group. So if you use anti-A, so type A, anti-B, type B, okay? So we have another one, is a monoclonal antibody anti-D. D basically means that rhesus blood group, another kind of antigen to show the positive or negative. Okay, so you can take another 1 minute to complete this table 12.8. Okay, based on the agglutination. Tick means agglutinations. Okay, so very simple in this case, anti-D give us whether the blood group is a positive or negative. Okay, if the agglutination means positive. Okay, so you use anti-A and anti-B. So anti-A positive, anti-B positive agglutination. So blood group AB positive. This one you can see the anti-A positive, B no, anti-D positive. So means that's A positive. And you do this, you can see the anti-B. So B but negative, B but positive. Can you see that? A negative, so it means that A but versus negative. O means that no agglutination, A and B. O means basically they don't have the anti-A and sorry, they don't have the antigen A, we don't have antigen B. So that's why no, but it's a positive. So this AB negative and nothing, everything O negative. So among all of them, which one is a universal donor? A universal donor, we say O, but O positive is not really. O positive is not really a universal donor if you compare to O negative. O negative right now is a universal donor. Basically, at the surface of the red blood cells, they don't have the antigen A, they don't have the antigen B, they don't have, okay, the antigen D, which is red rhesus, okay? So next, some monoclonal antibody, they have a significant success in treatments that involve modifying the immune response. So this monoclonal antibody, they are strazuzumabs. Can you see that? The drugs that end with the MAB, the name, they are monoclonal antibody. Okay, they are monoclonal antibody. Okay, so again, in this table, you do not need to memorize. Only one that you need to know is strazuzumab, or the commercial name called Herceptin. Commercial name called Herceptin is to treat breast cancer. Okay, is to treat breast cancer. So ipilimumab, infliximab, rituximab, those are, for example, ipilimumab, okay, for the melanoma, a type of skin cancer. Infliximab we use for rheumatoid arthritis, a kind of autoimmune disorder. Rituximab is inappropriate production of B cell, for example, leukemia or autoimmune disorder. So only one you need to know, strazosumab, which is a commercial name called Herceptin to treat breast cancer. Only one. Okay, so I don't go into detail because nothing much is very factual. So let us look at this strazosumab. Strazosumab also a commercial name called Herceptin is a humanized. Why we need the humanized mouse monoclonal antibody? Okay, can I see that? Humanized mouse monoclonal antibody is used in the treatments of some breast cancer. Now, why is it called some breast cancer? Breast cancer, you have certain category. So breast cancer cells, sometimes they express a certain receptor. Okay, so for strazosumab to be used, they must express a receptor, surface receptor called HER2 receptor. Okay, it's a receptor protein called HER2. Okay, so this HER2 is overexpressed in this breast cancer cell. So we need to check. If HER2 receptor is not overexpressed, other receptor, then strazosumab cannot be used. Strazosumab only can be used when HER2 receptor is overexpressed. Okay? So that's why it's some, not all. Okay? So it binds to a receptor. This strazosumab binds to a receptor protein called HER2 that is produced in the abnormal quantities in the cell surface membrane of some breast cancer cells. So the receptor protein is not unique to cancer cells because normal the normal breast cells also have this Herceptin, but the cell with between 10 to 100 times the usual number of these receptor number can be only in cancer cells. So it means that the normal cell may be 1 or 2, but cancer cell may be 100 times. Okay, so cell treated with strazosumab will undergo arrest during the G1 phase. So it means that they will stop at G1. Therefore, they cannot continue to proliferate. So we can stop the proliferations of the cancer. Okay, so now what actually happened here is HER2 receptor normally will receive the signal. When they receive the signal, they will transmit the signal to the nucleus so that the cell will grow and divide. Because the number of HER2 receptor is not huge enough, so it means that the chances for them to detect the signal normal. But if the HER2 positive, okay, breast or stomach cancer cells in this case, that didn't generate breast, okay, so when they have more receptor in this case, basically means they have higher chance to receive the signaling molecule. Once they receive the signaling molecule, they continue to proliferate. The cell will grow quickly. So now the idea here is we want to block the receptor by using the monoclonal antibody. This monoclonal antibody is strazosumab. So when strazosumab or Herceptin bind to this receptor, so there is no signal transmission, okay, because, okay, they bind already, so no signal transmissions, so the cell won't divide. So therefore, the cell will be arrested at G1 phase. Okay, so they won't continue, because we know that if you stop it at G1, it won't migrate to S phase, it don't migrate to G2, it won't carry out mitosis. So the breast cancer cells can be, what we call this, we limit their growth. Okay, but just now we talk about humanize. If we use the mouse monoclonal antibody for the treatment, this is a long-term treatment, it's not a 1 day, 2 days, then you can see that our immune system will, because the mouse monoclonal antibody for us will be the non-self antigen. We will produce immune response to get rid of this monoclonal antibody. So it means that the first treatment may be success, second treatment, third treatment actually will fail if we don't do humanized monoclonal antibody. So what I mean humanized monoclonal antibody? So generally you can see that when considering antibody for therapeutic purpose, not diagnosis, therapeutic means that for long-term using, it is important and mindful of the immunogenicity of the drugs. Because if you use the mice monoclonal antibody, rat monoclonal antibody, this monoclonal antibody will be treated as non-self antigen. So humanized antibody are antibody from non-human species whose protein sequence have been modified to increase the similarity to antibody variants produced naturally in human. Are you clear? Okay, so you can see that we try to reduce. If you use a mouse, all red color. So means that every single part can be detected as non-self antigen. If chimeric means that we remove this variable region from the mouse, add it into the human's antibody, then these two parts will be treated as the non-self antigen. Still, that is a dangerous step. Humanize basically means that we only choose particular regions, put it into the human's antibody. So in this case, you can see that it reduces the risk to be rejected by our human's immune system. And this is a human's antibody. Can you see that? The differences. So explain 3 advantages of using humanized monoclonal antibody for therapeutic purpose. So the immunogenicity of the humanized monoclonal antibody is reduced. Now, you cannot say no. We still have, okay? So because the immune response occur against the mouse antibody occur at a constant region, so now we remove the constant regions, so we reduce only, okay? So the human FC regions of the humanized monoclonal antibody allow FC receptor to recognize and bind the FC region of the antibody. So can you see that the FC regions of the antibody, it still remain as a human one. So if it remains as a human one, so means what? Means that the FC regions can be recognized by phagocyte, then opsonization can take place. Okay. Third, the plasma half-life of the humanized monoclonal antibody is significantly increased. Why? Because humanized monoclonal antibody are less likely to be removed from blood plasma and it is closely resemble human antibody. So basically, in this case, our immune system won't remove them a lot. So they remain in high concentration. So continuously give us the therapeutic effects here. Okay, so with this, I have done for the antibodies. Okay, so I stop the recording.